All right, today is one of the last parts we're going to be taking a look at is how gene technology is going to be involved with that of our genetics unit. We take a look at what gene technology is. If I just kind of shrink this a little bit, why is it important for us to actually know about and learn? What it really comes down to it's what you guys are going to be seeing as your future. All right, you can look at the Mayo Clinic. How have they advanced? We are now seeing what's called individualized medicine. We are doing medicine based upon you. But if we didn't know who and what makes you up, we wouldn't be able to treat you or treat any of the illnesses you might have. It really comes down to it's your future. All right. It's the world we're growing up in. We've got a lot of these things. We need to try to treat as much as we possibly can. Treat cancers. Try to improve medicines. All right. Now, if we move forward, we're taking a look at moving genes from one organism to another. That process of moving genes, transplanting genes from one organism to the next is called genetic engineering. All right, I engineer something that to be better or not. And a lot of the places we actually see genetic engineering being done is actually in cropland. Okay, cropland does a lot of genetic engineering. What that is doing is it's taking some of the good things from all sorts of areas, things that have been working well for a particular organism, and transplanting it into one that we might think is going to be beneficial for later. Now, take for an example corn, right? You got the corn all around the fields around here. Now, one of the things you might even see commercials on is being pesticide resistant, or animals won't eat it because of certain things that are going to be with inside of the actual genes of the corn. So that if I can spray it with certain pesticides, that corn won't die, but all the other pests that are around will. Okay. Other examples of things that we can do is we can make human proteins with the use of bacteria. Bacteria is one of those really cool organisms that we can actually manipulate. Now, the thing about bacteria, and you'll see here in a little bit, bacteria is um, those organisms that do not have a nucleus, but what they have inside of them is DNA, just like we have. But their DNA is called a plasmid. Now, what I just drew out to you is a DNA of a bacterium. Now, if I was to actually stretch that DNA out, it would be a nice big circle. In fact, it would probably be much larger than the cell itself. So that plasmid is that genetic material. But what's unique about the plasmids of DNA of bacteria is that all of the traits or genes, so these are certain genes that are within here, all of them get transcribed and translated. So we talked about in terms of humans, sometimes we have wasted letters, wasted material, things we don't copy. In bacteria, they use every single letter. So for example, what I want to do is I want to erase some of this. Now if I want to go back to, let's shrink this up a little bit and move this on. If I had our blue down, our blue cell, but all of a sudden I popped it open and I wanted to insert a new gene, I can do so. All right, and say this new gene can make it actually glow in the dark. We can manipulate that to work properly with the use of bacteria because it's going to copy and make everything. And if you take a look at this illustration that we said in the picture, right, we see it not only making insulin, we also have it as making clotting factors for people that have hemophilia, the inability to clot. We also use this to improve medicines, antibiotics to try to get it better. Now, most of the food we are eating, most of the food we eat is genetically modified. Now, sometimes some of us are going to say that's awful, and in fact, in some parts we may not know how bad it's actually doing for us, but we do know that we are improving, according to us, our tastes as well as appeal. Now, when you go to the grocery store, are you buying the fruit that's all brown and kind of mushy or mushy looking? Probably not. You want to buy the biggest, the brightest, the nicest looking. So what we do is we genetically modify it so it looks like that. So it's pleasing not only to your stomach, but it's also pleasing to your eye. It does the same thing internally, but modifying it, it's making you pick it first. Alright, so some of the ways that we can genetically modify, we can pick and choose, as you see in this illustration on the right-hand side, pick and choose the genes we want to put in. We do that by manipulating DNA in certain ways. Okay, we can cut and join things together by the use of what are called restriction enzymes. Okay, restriction enzymes are actually going to find a particular sequence of letters. So it is restricted to a sequence of letters. So if I have letters A, T, 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 C, C, G, this enzyme that I come in is specific for that site. It has to find that site. If it doesn't find it, it won't cut it. So that's what a restriction enzyme is used for. Second thing that you see on here is PCR amplification. What PCR is, it's a fancy term. It's called polymerase. We've heard that before. Polymerase chain reaction. What that really does is it's going to put down a whole bunch of copies of DNA. I can take a small section of DNA, and if I want, well, let's actually write it in DNA form, something like that. I can take that small section of DNA, and I want to not just make one strand of it, I want to make 100,000 copies of it, 100 times the amount, or you name it. How many more DNA do I want? You see that a lot in professions like CSIs, where they have one droplet of blood. If they screw up in that one droplet of blood, they screwed up their test. So if I can amplify that amount of DNA and have multiple sets of it, I can have plenty of tests that I can run and not have to worry about losing any DNA. Okay, more techniques that you might see: cloning, making identical copies, DNA fingerprinting. This is getting what your DNA is. Notice that you are different from the person sitting next to you. The only people that have the same identical DNA are going to be identical twins coming from the same egg and sperm. Everybody else's DNA is going to be different and unique to them. Now, how do they figure that out? They can use that by sequencing, identifying what it is that's going to be within them. All right, so what do I do in order to some of those techniques? I can start to separate out DNA based upon size. Now, in this example, you see an illustration on DNA fingerprinting based upon size of genes that I've separated out. Now, the process of actually separating out those genes is done through what's called gel electrophoresis. So if you see what's happening here, I've got this big well. I've got this tray here, and I've got these wells that I'm going to put somebody's samples in. Okay? Now, if I put DNA into those little samples, some sort of a solution in those little samples, I want to start to separate out all of the different genes or strands that I have in there. Now, how do I do it? DNA is negative charge. So what I want to do is send an electrical current through it. Now, in order to do that, we got to remember how do charges work. Negatives are attracted to positives. So if I separate this out a little bit, what I'm going to do is I'm going to add down here a positive charge and a negative charge. Basically, we put an electrical volt on it. And if I have negatives here, negative and DNA is negative, they start to repel each other. So the DNA starts to work down towards the positive region. Now, the faster or the smaller the sections of DNA are, the faster they travel. So in a certain amount of time, the smallest sections will reach all the way down to the bottom, where some of the larger ones are going to be further up. So if we take a look at our illustration that we have here, this is all done in one section of time. But this section down there is much smaller in size than the guys that are up there. Same here. Those guys are smaller than the ones up top. Here's another illustration that just shows that same thing. The larger ones are up top, the smaller ones are then onto the bottom. Okay, larger, smaller will be following this way down. Here's our, again, PCR. The human genome has 3.2 billion base pairs, A's to T's, C's to G's. What we want to be able to do is we want to take a small amount of it and amplify it. Amplify it means make many of it through the use of this polymerase. We saw that in DNA and RNA polymerase, making the copies. Application for it, when do we use it? We mentioned in forensic, we're also doing it in genetic testing. Now, in a couple weeks, we're actually going to do some PCR amplification and see how this does work and amplify some DNA, in particular bacterial DNA, and see the amount of genetic material. And what we will actually do is we will take that genetic material and we will cut it up with restriction enzymes. So we have certain sections of DNA, and then we will separate out that DNA based on base pair length. And so we will do not only PCR reactions, but we will also be doing gel electrophoresis. Moving our way into slide number 9, here's an example of DNA fingerprinting, which is just shown before. How you can identify if there's somebody wrong. Now, in this case of how a CSI might utilize this, if you take a look at the evidence, we've got all of these were original wells that they put the genetic material into. Here we have a victim. Okay, you have somebody's victim. Now, what they found in the actual female during, for example, a rape case was the sperm DNA. They found some sperm in there. All right, they separated out the genes and identified that there were two sections of genes. Now they end up having two suspects. Suspect 2, when they looked at their sperm, that was right there, versus suspect 1 was there. So just by looking at that section, you can identify that suspect 1 was our culprit. Okay. So there's another example that's shown there based on specimens. Now, determine the order of the DNA. Identify the approximately 20 to 25,000 genes in the human DNA. Store this information in databases. We have a process that is using all of our genetic information. We're storing it in a big data warehouse. What we want to do with that is then identify what these particular genes do. So if we see here that identify approximately 20 to 25,000 genes in the human body, what are we doing with those genes? Okay, what do they code for? How do they make us human? All right, and then maybe there's certain ones of these genes that actually give us faulty tendencies. For example, the BRCA2 gene for breast cancer. We may not want that gene, so what is it that we can do to get rid of that gene, or maybe not have that gene? Okay. I'm sure you've done debates before on cloning. Cloning, what you end up getting is an identical copy of oneself. You're taking the genetic material, and you get what was in the parent cell in the daughter cells as well. Um, Ethical debates behind that, yes, blame God, human cloning. One way is people think that they can do is if they feel that they're superior, they can end up being genetically superior to other individuals based upon getting things that we want out, certain hair colors, eye colors, you name it. We're getting to that point where we can do this. And the last thing that we also see being used right now, if we have genetic diseases caused by some sort of missing or defective gene, we can start to take out some of the patient's cells, maybe blood cells or body cells, and we can insert a good copy back into the gene. And hopefully the body's ability to take that gene back in and put the good things back into their body. So really, just like if you're doing PT, physical therapy, trying to heal yourself, and say if you have a leg injury or a knee injury, whatever it may be, healing yourself, here, we're trying to heal the genes. We're trying to get them back to doing their normal functions. Okay. Now this is the quick illustration on biotechnology. I hope this helps, and we will be seeing you in the next few days.